2002, the scientific world witnessed something remarkable. A modest engineer from Japan, working quietly in a corporate laboratory, received the Nobel Prize in chemistry. His name was Koichi Tanaka, and his invention would transform how scientists study the building blocks of life itself. Born in 1959 in Toyama, Japan, Tanaka was not your typical Nobel laureate. He held only a bachelor's degree in electrical engineering and had spent his entire career at Shimadzu Corporation, a scientific instruments company. While many laureates emerged from prestigious universities with decades of published research, Tanaka's path was different. He was a company engineer solving practical problems. Before Tanaka's breakthrough, scientists faced a fundamental challenge when studying large biological molecules, particularly proteins. These molecules are essential to virtually every process in living organisms, from digestion to immune response. Understanding their structure and composition is crucial for developing new medicines and treatments. However, analyzing these delicate giants presented a seemingly insurmountable problem. Traditional mass spectrometry, the gold standard for identifying molecules, worked beautifully for small, stable compounds. The technique involves ionizing molecules and measuring their mass, but proteins are enormous compared to simple chemicals, and they are fragile. The intense energy required to vaporize and ionize them would tear them apart, like trying to weigh a butterfly by throwing it into a furnace. Tanaka's innovation, soft laser desorption, elegantly solved this problem. The technique uses a laser to gently lift large molecules into the air without destroying them. The key lies in mixing the protein sample with a special matrix, a substance that absorbs the laser energy. When the laser strikes, the matrix absorbs most of the energy and evaporates, carrying the intact protein molecules along with it, like passengers on a gentle breeze. The story of Tanaka's discovery carries an element of serendipity that often accompanies great scientific advances. In 1987, while experimenting with different matrix materials, Tanaka made what he initially thought was a mistake. He accidentally mixed glycerol with ultrafine metal powder. Rather than discarding the contaminated sample, he decided to test it anyway. The results were extraordinary. The metal particles enhanced the ionization process, allowing even larger proteins to be analyzed intact. This seemingly simple addition of metal powder to the matrix revolutionized the field. Suddenly, scientists could identify proteins that were previously impossible to analyze. The technique opened doors across multiple disciplines. Medical researchers could identify disease markers in blood samples. Pharmaceutical companies could verify the purity and structure of protein-based drugs. Biologists could map the proteins expressed by different genes. The impact extended far beyond the laboratory. Soft laser desorption became instrumental in proteomics, the large-scale study of proteins. It accelerated drug discovery, improved disease diagnosis, and enhanced our understanding of how cells function at the molecular level. When the Nobel Committee announced Tanaka as a co-recipient of the 2002 Nobel Prize in Chemistry, many in the scientific community were surprised. When the Nobel Committee announced Tanaka as a co-recipient of the 2002 Nobel Prize in Chemistry, many in the scientific community were surprised. Here was a corporate researcher with minimal publications, sharing the stage with established academics. Tanaka's work had fundamentally changed how science is conducted. Tanaka himself was reportedly shocked by the news, learning about his Nobel Prize from a television broadcast. His humble response and genuine surprise endeared him to many. He represented a different kind of scientist, one driven by curiosity and practical problem-solving rather than academic ambition. The soft laser desorption technique, now commonly known as matrix-assisted laser desorption ionization, or MALDI, has become a standard tool in laboratories worldwide. Thousands of research papers rely on this method every year. It stands as a testament to how a single innovation, born from curiosity and a willingness to examine mistakes, can ripple across entire fields of study. Koichi Tanaka's journey from electrical engineering graduate to Nobel laureate reminds us that groundbreaking discoveries can emerge from unexpected places. His technique gave scientists a new lens through which to view the molecular machinery of life, transforming a butterfly too delicate to touch into something we can finally understand.